Thank you so much for having me here today. I'm here to talk about some of the work that my lab has been doing in using lipid nanoparticles to deliver a type of drug called siRNA. So before we get started on the specific topic, I just wanted to give a bit of an overview of my lab. We focus on drug delivery, which involves altering the transport properties of different types of therapeutic molecules so that they can take effect in patients in the least invasive means as possible. And so I would say there are two major types of drug delivery that are done today. One involves the delivery of FDA-approved molecules that may be administered in ways that are less friendly to patients. So any kind of an injectable medication we could take and we could try to deliver it perhaps in an oral means. So that's what half of my lab does, developing ways of delivering proteins and peptides and other macromolecules orally. You can think of insulin as an example of a drug where we might, instead of injecting patients 3 times a day, prefer to take that orally. The other type of drug delivery being done today involves taking drugs that are not FDA approved and developing means by which we can introduce them into the body where they can take therapeutic effect. And so that's what I'll be speaking about today, focusing on injectable ways of delivering genetic material, RNA molecules, and specifically siRNA, which stands for short interfering RNA. And I'll be telling you how we can package this drug up and administer it such that it takes effect in different organs and cells throughout the body, including the liver, immune cells, and in different types of cancer. So before I get into the specifics of what we do in my lab, I wanted to tell you a little bit about the biological pathway that this drug takes advantage of. It's called the RNA interference pathway, and it was discovered by biologists in 1998. And it was awarded the Nobel Prize as quickly as 2006. So that's only 8 years between discovery and Nobel Prize. It really underscores a lot of the potential that this type of drug has, not only for therapeutic purposes, but also because it can aid in biological discovery and understanding. So what this pathway involves is this drug molecule called short interfering RNA. It's about 21 base pairs in length. And the trick for engineers is to figure out how to get this drug molecule into the cytoplasm, into the interior of the cell, where it can then interact with the RNA interference machinery. And so there's this enzyme called RISC that if siRNA is able to get inside, it will bind to the siRNA, it will unwind it, it will mediate the cleavage of the sense strand, and it will take the remaining strand and it will escort it around the mRNA genome, searching for a piece of mRNA that is complementary to it. And should it find it, it will mediate the cleavage of that piece of mRNA, silencing the gene and silencing the production of any protein associated with the gene. And so by delivering a very sequence-specific type of RNA, we have the ability to turn off the production or to reduce the production of nearly any protein that's expressed in the body. And so this really has tremendous therapeutic potential, and we can think about using siRNA to treat many different types of disease, including cancer. So you think about different types of proteins that are overexpressed in cancer cells that are forcing the cells to divide and to push their cell cycle forward. We can go in and we can try to lower the expression of those proteins. In the case of viruses, we have viruses that go in and they hijack some of the human machinery within the cells to help replicate these viruses. And so we can use RNA interference to turn down the production of these proteins that the viruses hijack. And then lastly, you might think about hereditary disorders, high cholesterol being a great example. We can go into the liver and we can reduce the expression of proteins that help to produce too much of that cholesterol. And so really there is a tremendous amount of potential here, but the challenge is in delivery, getting this siRNA inside of the cell. And this is because our siRNA is large. It's about 13,000 grams per mole. It's negatively charged, which means it does not readily cross cell membranes. And it also has really lousy biodistribution properties in vivo, meaning that if we were to simply inject it in its naked form into the bloodstream, it would be cleared very rapidly by the liver. So what we need is to think about some kind of engineering strategy to package up this siRNA drug that will overcome some of the barriers within the body and take it to the cells where it needs to go. And so if we were to think about some of the barriers that a delivery system would need to overcome in the body, we could think about some of the following. So once we inject this particle into the bloodstream, the first thing that typically happens is that the macrophages, the immune cells within our body, are interested in gobbling up this particle. So our particle needs to be able to avoid phagocytosis, macrophage uptake inside of the bloodstream. It then needs to be able to exit the bloodstream into the extracellular matrix, which it needs to diffuse across to the vicinity of the target cell, whatever organ that might be that we're interested in treating. And once it's there, it needs to somehow gain entry into the cell. This is typically done through a process called endocytosis, which essentially forms a little wall around our nanoparticle. It's the cell's way of sampling its extracellular environment, and it's generally not sure if it actually wants to keep whatever is inside of that endocytic vesicle. The cell generally does not want this lipid nanoparticle inside of it, and so it would often degrade that particle if the particle cannot escape. So one of the most important parts of this process is encouraging what we call endosomal escape, to have our particle exit this membrane vesicle and get out into the cytoplasm, where it can interact finally with that RNA interference machinery I spoke of. So one of the things that we asked a number of years ago was whether or not we could develop a potent siRNA delivery system that importantly would be degradable. And the degradability is important because siRNA therapeutics, when you inject them, they're going to reduce a protein, but they're only going to do so for a limited amount of time, depending on the protein and depending on the organ type that we're going after. And so if you're really going to administer this long term, you're going to have to dose someone again and again and again with these therapeutics. So it's very important that we work with a material that's going to be biodegradable. So we designed materials that we call lipidoids. They're lipid-like materials, and we designed them to mimic traditional lipids that have been used for gene transfection in the past. And so DOTMA is an example of a cationic, traditional two-tailed lipid. It has some unsaturation. And this material would be used to form nanoparticles that you can think of as liposomes. They have a lipid bilayer containing an aqueous core where you could put a hydrophilic molecule like siRNA. And so these have been used in the past, but they have numerous challenges, toxicity being one of them associated with this cationic, very positively charged molecule. And so we wanted to come up with a chemistry that would give us this simple liposomal structure, but perhaps offer some different efficacy levels without the toxicity. And so we would react alkylamine molecules, so we have a primary amine in this case, with alkyl acrylate tails. You can mix them together in a scintillation vial without any solvent. You can add a stir bar, put them at 90 degrees C for a couple of days, and out the other side comes this lipidoid molecule. In this case, we've designed it such that it has two tails because we have a single amine here to start with. But depending on this amine chemistry, we could have two tails, we could have anywhere up to 8 tails. And so it doesn't necessarily have to look like your traditional two-tailed lipidoid. And importantly then, these materials have that degradable functionality in them in the form of these esters. And so under physiological conditions, water will help to reduce this bond to an acid and an alcohol, which helps these materials to clear out of the body. So lipidoids are really nice in that they electrostatically interact with siRNA, which helps the formation of nanoparticles for them to be carried. And so the ones that work well, the lipidoids that work well, they tend to have a pKa usually in the range of about 6. And what that means is that if we drop below that pKa in pH, so if we were to formulate these materials at a reduced pH, these materials will take on a positive charge. siRNA is extremely negatively charged, and so then we get this really nice electrostatic interaction between our siRNA and our lipidoid. Then, beautifully, when we're interested in delivering this, we're going to be at physiological conditions, which is a pH of 7.3. When we bring the pH up, we actually lose all of this positive charge. But these materials are now contained in a nanoparticle. So we wind up having a neutrally charged nanoparticle, which will be important in reducing some of the long-term toxicity that we've seen with other systems. So we formulate our materials into nanoparticles either by hand, you can do this by simple pipetting, or through an inline microfluidic device, in which we introduce an siRNA stream, shown here in red. That's an aqueous stream, with the lipid mixture stream, which is dissolved in ethanol. These will form nanoparticles, and then we rapidly dilute them with phosphate buffered saline, or PBS, to help them prevent aggregation. And then our lipid nanoparticles, in addition to containing the lipidoid, which is the active lipid component, we also have cholesterol, which is added for stability and rigidity of our particle, as well as DSPC, which is a helper lipid, and polyethylene glycol, which helps to avoid phagocytosis, that macrophage uptake in the body. And these particles, if we look at them under cryo-TEM microscope, we see that they're about 80 to 100 nanometers in diameter. So what we did was we synthesized a library of 1,400 of these degradable lipidoids. And we were able to make so many because the chemistry is quite straightforward. So the way we did this was we took 280 different amine chemistries and we combinatorially reacted them with 5 different tails that varied in the carbon chain length between 10 and 14. So when we did this, we might say mix together a tail that we might call O10, so that's a carbon tail, with an amine that we might call 25, and then we would make a lipidoid that we would call 25O10. A lot of people ask me, why high throughput screening? A lot of other people in the field use rational design to design materials that they think would make sense, why the chemistry would make sense to overcome some of these delivery barriers. And so why should we test so many materials? And the answer that I give is that we often don't understand the biology that we're actually trying to overcome. So when I talked about the challenges that we were trying to overcome in the body before, when we talk about phagocytosis and avoiding macrophage uptake, when we talk about getting things specifically to the liver or getting them into the cell, these are only the barriers that we can imagine. There are probably other barriers that we don't understand, and we also don't understand what specific chemistries are needed to induce these functionalities, let alone all of them together. And so by doing high throughput screening gives us the best possible chance of identifying chemistries that are able to overcome all of these challenges. Then we can step back and ask ourselves, how did this chemistry actually perform the function, and what can we learn from that? So we took these 1,400 lipid nanoparticles and we first asked whether or not they could deliver siRNA in cell culture. And so to do this, we took our particles and we applied them in high throughput format to 96 well plates using what we call dual HeLa cells. So these are HeLa cells that have been stably modified to express 2 different types of luciferase, which is essentially a type of light. And the 2 types of luciferase are firefly luciferase and ranilla. And so we include an siRNA that is specific for only the firefly. So we want to silence the firefly and ideally leave this ranilla alone. So what we would expect to see for a material that works well is a reduction in the firefly expression without seeing anything happen to our ranilla signal. If we did see that ranilla signal go down, that would be indicative of silencing that is not actually gene specific. It would be indicative of some type of toxicity. And so this system has a really nice internal control that we can use to evaluate our results. So when we took these materials and we looked at how they performed in this dual HeLa cell culture, what we're looking at here is relative luciferase activity on our y-axis. Okay, so 100% activity, or 1.0, that means no silencing. It means that there's as much of the protein there as there normally is. And then we're looking for reductions in that. And that's for our 1,400 different compounds here at a particular dose. And so what we can see here is that while most of the materials in black, they did not induce very good silencing, we did have a good number here, about 7% of our library, that gave us 50% silencing of luciferase, the firefly luciferase, or better. So this was promising, and we took a number of these materials in red, and we decided to put them in an animal and see if they could work there as well. And so in this case, we were interested in silencing a protein made in the liver. It's called factor VII, and it's a blood clotting factor that is secreted into the bloodstream. And so it's very easy to measure because you can just take a blood sample from the animal. So we would take our lipid nanoparticles containing siRNA specific for factor VII, and we would inject them into the tail vein of the animal. And 2 to 3 days later, we would take blood samples, which we would quantify for the amount of factor VII. So here the graph we're looking at is our relative factor VII activity on our y-axis. And again, full expression, no silencing is at 1.0, and we're looking for reductions. And in this case, we tested about 100 different types of lipid nanoparticles. So what we see once again is that a lot of these particles are not able to overcome the challenges of the full mouse body. But we have about 15 materials here shown in red that induced a full 100% silencing or so of our factor VII in these animals. So with a little more work, we took those 15 materials and we tried to find the best of those. And this data shows about the best that we can do with our lipid nanoparticles. What we see here for factor VII activity is that this particular lipid nanoparticle is dose responsive and that we can see silencing for a period of about 2 weeks. So if we look at our red curve right here, this is the highest dose administered in this case of 0.1 mg per kg. We see that we have about 100% silencing to start with, and it returns to baseline over a period of about 18 weeks. When we reduce the dose to 0.03 and then further to 0.01, we see reductions in activity, and that's what dose response means. And the duration also lessens. And so we were excited because at the time, this was among the best delivery systems in the world as far as potency was concerned. When it comes to potency, the best thing about having a material that can be quite effective is that we can go in and we can potentially silence more than one gene. A lot of diseases have multiple proteins that are responsible for that disease action. And so it would be wonderful if we had a delivery system that with a single injection, we could knock down not just one protein, but multiple. And so that's exactly what we could do with this particular system. Here we're looking at 5 different proteins that their activity starts at 100%, and as we increase the dose of these siRNAs, look at the silencing that we're able to achieve. And this is all with a single injection of these particles containing 5 different types of siRNA. And so this is very powerful and will allow us to move forward at the treatment of a number of different diseases. So our lead lipid nanoparticle, as we might expect, it accumulates in the liver. If it's able to silence a protein in the liver, we expect that it would be delivered there. So this is some images taken where we injected siRNA that had been labeled with a fluorophore called Cy5.5. And so without any delivery vehicle, if we were to inject this into the mouse, you see there's very minimal accumulation here in the liver. However, we have very quick clearance through the kidneys. And these are 2 different types of images that confirm the same event. So in this case, we see that we have a lot of accumulation through the kidney, but it's a totally different story when we put it inside of our lipid nanoparticle. Now look at how we've shifted the biodistribution. We've minimized accumulation within our kidney at 1 hour, and instead we've shifted that biodistribution to the liver, which is our therapeutic organ of interest, as well as the spleen. We also wanted to make sure, in addition to having good efficacy, we want to make sure that we don't have toxicity. And there are a number of different ways that you can check for this. So one of the ways that we looked at was using histology to look at whether or not our particles could cause necrosis in the liver. So this is an H&E stain of an untreated mouse liver. It's healthy. Here's a very high dose of our degradable lipid nanoparticle. The difference in color doesn't matter. What matters is that the shape of the cells is about the same. So this is a 15 mg per kg dose, which is quite high. In contrast, this is our lead lipid nanoparticle that is not degradable. And what you see here are regions of necrosis and liver damage throughout at the same dose. And so there really is an advantage here in delivering a vehicle that is degradable. So delivering siRNA to the liver is great. Some would even say it's easy. It's not easy, it's easier, because it's one of the organs that's interested typically in taking up particles and clearing them from the body. So we were pleased that we were able to do this, but at the same time we wondered if we could induce silencing in a more challenging gene target. And at the time I was thinking, we're injecting these materials into the bloodstream. So how about a cell target that's in the bloodstream, since that's what would see these particles first? Blood cells can be pretty difficult to deliver siRNA to, but I've been particularly interested in looking at blood cancers. Blood cancers are going to be rapidly dividing, and that rapid division actually creates an extra opportunity to try to introduce materials into the cell, because the cells become a little more vulnerable when they divide. So my lab has been looking at using our lipid nanoparticles to treat a type of lymphoma called mantle cell lymphoma. This is one of about 14 subtypes of B-cell non-Hodgkin lymphoma, and it's typically treated with a combination of chemotherapy, immunotherapy, and for younger patients that are healthy, autologous stem cell transplantation. But unfortunately, it's still one of the most deadly types of blood cancer that you can get. And most patients are going to relapse very quickly. It's simply an aggressive cancer that keeps coming back. And the problem is that chemotherapy causes damage to the heart. And so over time, patients simply cannot handle these repeat doses of chemotherapy. You have to reduce administration of the drug, and patients typically succumb within about 5 years of diagnosis. And so there's an opportunity here to try to introduce a new type of drug to improve outcomes for these patients. So we think that RNA interference therapy could do quite a good job here, and it's because in part of why mantle cell lymphoma occurs in the first place. And so if we were to look at the genes involved in the progression of this disease, one of the most important ones is a protein called cyclin D1. This here is a protein that causes the cell cycle to move forward. It encourages the cell to divide. And normally it occurs on chromosome 11, where it does its appropriate thing. However, in the case of mantle cell lymphoma, what happens is that it gets swapped with chromosome 14. And it gets placed downstream of what's called an Ig heavy chain enhancer, which is something that causes the gene downstream of it to get produced a lot. And so in this case, we're going to have much too much of our cyclin D1, which causes the cells to divide much too rapidly, and thus we start cancer. So because we have this very specific gene that's been implicated in the disease, as well as several others, we know that RNA interference has particular potential in helping to treat this type of disease. So we took our 1400 lipid nanoparticles again, and we wanted to ask, can they deliver siRNA to this very difficult type of B-cell cancer? So for our 1400 particles, we ran some experiments where we took lymphoma cells that were modified. They expressed a fluorescent protein called GFP. Okay, so we're looking at relative GFP expression here on our y-axis. And again, we're looking for reductions in that activity for materials that are working well. On the x-axis, we're looking at the relative viability following delivery of this particle. Now, it's important to understand that after delivery of just our particle, we don't want any toxicity, because if we see toxicity, that means that the delivery vehicle itself is causing toxicity, and that's not acceptable. We don't want our delivery vehicle to kill every cell within the body, just the cancer cells. So the group that we ultimately were interested in are these red points right here, because they were able to induce silencing without inducing toxicity in these cells. And it's interesting to compare how difficult it is to deliver siRNA in these lymphoma cells compared to the HeLa cells from before. So this is a 2% hit rate at a dose of 100 nanomolar in our lymphoma, compared to a 7% hit rate at 40 nanomolar for our HeLa cells. And so I would estimate it's approximately 10 times more difficult to deliver siRNA to these lymphoma cells than to HeLa cells. So then the question became, okay, we can deliver siRNA, but can we actually silence the therapeutic targets for mantle cell lymphoma? One of which is MCL1. So this is a protein I haven't talked about before. This is a protein that prevents the cells from undergoing a process called apoptosis. Apoptosis is when the cell essentially commits suicide when something has gone wrong. So normally cancer cells would get the signal. They would say something has gone wrong here, and they would begin to self-destruct. However, when there's too much of this protein around, they don't do that. So we would be interested in reducing the expression of this particular gene. So what we wanted to do was to test our siRNA lipid nanoparticles in a couple of different types of mantle cell lymphoma cells. And the first type is called GECA1 cells. I call these easy cells to deliver to, not because they're easy, they're not at all, but they represent standard disease, and they are easier than some of the patients that present with very complex, more difficult disease. So this is more of a standard type of mantle cell lymphoma. So what we found when we looked at gene silencing of MCL1 was what we would expect based on previous data, that our lipid nanoparticles were doing a very nice job of reducing this relative MCL1 expression, and that's the curve in red, whereas our controls, as they should, were not doing much of anything. So then the question was, okay, so we've silenced, but does that actually result in any effect? So when we looked at the number of cells undergoing apoptosis in response to this treatment, we saw that whereas the untreated cells, they're apoptosing about 5% of the time, after gene silencing, they're apoptosing 30–35% of the time. Okay, so we have a lot more of these cells beginning to die in response to therapy. And I think it's very important in cancer therapeutics to consider not trying to cure one particular patient's cancer. And that's what we would have if we kind of stopped here. So we also wanted to make sure that we could use these lipid nanoparticles to treat another type of mantle cell lymphoma. MAVR1 represents a very difficult, very aggressive form of mantle cell lymphoma. We didn't expect the results to be as good, and the silencing itself is more modest. We have about 50% silencing here over a period of about 5 days compared to 7 or 8 days at 70% silencing with GECA1. However, the good news was that when we looked at apoptosis following therapy, we also had about 30% of these cells undergoing apoptosis. So this was promising that we could have therapeutic effect in multiple cell types. In addition to silencing MCL1, we're able to silence other genes that are important in the progression of mantle cell lymphoma, including BCL2, which is another anti-apoptotic protein. It works very similarly to MCL1. So in this case, with a 100 nanomolar dose of our siRNA, we're able to induce about 80% gene silencing, and our results are dose responsive. And the same thing with cyclin D1. This is the protein responsible for extra proliferation of the cell. So we're able to silence this one too. So we thought this was great. We now have 3 therapeutic proteins that we can try to go after, and we wondered, well, if we deliver these in combination, would that give us even better effect? And so we looked at these cocktails, and we found that they do induce a lot of apoptosis. So here on the y-axis, we're looking at the number of cells undergoing apoptosis. You can see that our untreated cells and these control cells that have received just the delivery vehicle without any RNA, they're undergoing apoptosis about 5% of the time. When we deliver siRNAs against any one of these 3 targets, you can see then we have about 30–35% apoptosis. When we deliver 2, we see improvements in that level of apoptosis. And with 3, we have a full 75% of these cells undergoing apoptosis, which is terrific. And results are statistically significant as well. And so we were really encouraged by this because this therapy is not meant to be used by itself. This would be used in conjunction with the chemotherapy and immunotherapy that's commonplace today. And so with this added tool, we're hopeful that we can have extra impact on our patients. So in a very preliminary animal experiment, we took these siRNA cocktails and we looked at whether or not they could inhibit tumor progression in an animal. So here on the y-axis, we're looking at our relative tumor volume in an animal as a function of time. Okay, and 0 days starts when the tumor is palpable. And one of the very important decisions that I think a cancer researcher needs to make is when do you actually start treating your mouse after it's been diagnosed in this case, or do you start treating before? Personally, I think it's more robust to start treatment after we know the mouse has cancer, because we can never treat patients before they're actually diagnosed. So here we've treated these animals on day 3, 5, and 7. And what you can see is that our purple and our red curves, our control and our untreated groups, these tumors are growing quite rapidly. Whereas our siRNA cocktail, when we delivered them intravenously, IV, or intratumorally, directly into the tumor, that's the green, you can see that tumor growth is reduced significantly over the course of 8 to 9 days. So this is encouraging, and we're looking forward to continuing these animal experiments. So today I've told you about our platform technology in my lab called lipid nanoparticles. They have a degradable chemistry, which is excellent in reducing the long-term toxicity associated with some of these drugs. They have the ability to be targeted. I did not talk about that today, but if we're interested in targeting them to specific types of cells, we can do that. And they also have a versatile drug cargo. I talked about siRNA today, but we can also use these materials to deliver other types of RNA, including mRNA, as well as proteins and small molecule drugs. And we can use them to deliver into multiple targets throughout the body, including the liver, blood cells, cancer, and some work I did not talk about today, the intestinal epithelium, skin cells, and even brain cells. And so with that, I'd like to thank you for your time and attention. And I'd like to thank the people involved in the work in my lab, especially Chris Knapp, who has spearheaded a lot of the mantle cell lymphoma work, as well as our collaborator, Dr. John Lister. And I'd also like to thank the following agencies for funding. Thank you very much, and I'll take any questions.